I started off in this as an academic physician. I was looking after children with very severe immune deficiencies. And actually what we found was the children would either die of their disease, or if there was a bone marrow transplant, then that could have side effects and they would die from some of the complications of that. And so really, this was now stretching back over 25 years ago, and in the very nascent era of gene therapy, and the feeling that actually using the child's own cells would be better than using somebody else's cells. And we had to then work out how to put genes into those cells safely and effectively in order to correct that disease. And so with a colleague at UCL and Great Ormond Street Hospital, we conducted some of the first trials to show that gene therapy could actually correct a monogenic disease, and that was in these forms of severe combined immunodeficiency. And that was a very powerful moment for us to see that effect and to see patients recovering from their disease through this gene therapy approach. And we built the expertise to be able to deliver that for not just those severe immune deficiencies, but for other disorders as well. Now, latterly, as we've seen that high level of efficacy and that high level of safety, we really wanted to take this out of the academic setting and to make it into something that could be used by children worldwide. So an approved and standard medicine. We were having children, for example, referred to us from all over Europe. We even had a child coming from Australia to be able to access treatment. And so really felt strongly this needed to be something that could be delivered on a global basis. And we needed a commercial entity in order to be able to put in all the infrastructure to make that happen. And that's how Orchard came about. And that's why I, having spent many, many years in both academia and in the clinical setting, have taken on a role as CSO at Orchard in order to be able to deliver these medicines for these various different diseases. So we've focused on a number of different conditions, and we've broadly broken those down into immune deficiencies, neurometabolic disorders, and hemoglobinopathies. So for immune deficiencies, for most of these conditions, the only standard of care is a bone marrow transplant, so taking somebody else's cells and giving it to those patients. And when you do that, you run the risk of graft versus host disease, where the donor cells see the host as different and can cause all kinds of problems. Now, what we want to do is to use the patient's own cells, and so we think that will be a much safer procedure. And so we've been able to do that now for 3 different disorders in our portfolio. So there's ADA-SCID, chronic granulomatous disease, and Wiskott-Aldrich syndrome. So each of those has now been in clinic using this autologous HSC approach, and all of these cases we've seen the potential for correction, long-term correction of the underlying disease using this approach. In the neurometabolic disorders, that was the other reason, the other franchise is in neurometabolic disorders. Actually, for most of these conditions, there's really no standard of care. But the use of HSCs really offers a unique approach to correcting these disorders. And that is because HSCs, a subpopulation, have the potential to cross the blood-brain barrier and become microglia in the brain. So if you can introduce a gene into those HSCs, you can deliver a gene to the brain. And so the way this works is that for some of these defects, there is an enzyme that is missing. That enzyme leads to the buildup of certain substrates in neurons, which causes neurodegeneration. The way to address this using this approach is to overexpress the enzyme in HSCs. That subpopulation crosses the blood-brain barrier, becomes microglia, and overexpresses that enzyme in the brain, which is then taken up by neurons and then prevents the substrate accumulation and the neurodegeneration. And we've now, for a condition called metachromatic leukodystrophy, or MLD, we've seen long-term correction of the disease with correction of motor and cognitive problems as a result of using this approach. And this is for a disease which is almost universally fatal over time, and in which there is no other treatment option available.